who is Maria Ruesseler, who's going to talk to us about evidence accumulation in a new .motion task. Zara, I think I need you to stop sharing your screen so that Maria can start sharing hers. Brilliant. Thank you very much.
Okay.
Yeah, so take it away, Maria.
Yes. Yeah, thank you, Dan, for the introduction. Hi, everyone. Thank you for your interest in my talk. I am Maria, and I work with Lawrence Hunt, and I will be talking about a project that I did during my PhD. And this is about how humans adapt the integration kernel to different timescales of evidence integration. So to give you like an example, if you think about everyday life and you might be using your bike, going to work, you will have to make decisions that are probably based on maybe a few minutes or even seconds of evidence, for example, to switch lanes. So this is a very short time scale. In contrast, if you maybe want to sell stocks, you probably want to think about that and consider evidence of several days or maybe even weeks. So how are we measuring this in the brain? Like Sarah, my task paradigm is a random .motion paradigm. And as you know, in the past, these paradigms have been consisted of discrete trials in which mostly the motion coherence stays constant. However, in my task, the dots change their motion direction continuously for 5 minutes straight. And this is a small video. I hope it works because sometimes Zoom will probably drop frames, but this is basically what participants will see. They will see these continuously moving dots, and then they will have to detect these stable phases in which the dots will more often move to one direction than to another. These stable phases are indicated by the fixation dot in the center turning white. So obviously, this is just for you guys so that you get a feel for the task. In the real experiment, participants won't get any cues about this. But importantly, participants have to pay attention to past motion directions to solve this task. This is a schematic of the task, and this black line, this is the signal, and this can be either in a baseline period or in a trial period every time the signal deviates from the baseline. So for example, here it goes down. That would be a response to the left, because here on average, we look at those and those, the motion goes on average more to the left than to the right. So now this is not everything. We thought that if we manipulate the trial period, meaning that we ask how does the integration kernel of participants change if the trials are either long or short, or based on how often they are they frequent, or are they rare? This is shown here in this schematic, and it's a 2x2 design. And then we would expect that the integration kernel shown on the right would change accordingly, and it would be how far back in time would you consider evidence, and how much evidence in total do you need to make a decision. So now we need to think about how do we analyze this data, and how do we get these integration kernels from the behavior, but also from EEG out of this data. And for this, the noise is very, very important. So the noise is this gray line here that wiggles around the signal, and that's the actual coherence that participants see on each frame. And very importantly, I have complete control over this noise, and I know exactly what happens on each frame. And this variability in the noise is, in the end, what allows me to analyze the integration kernel in the behavior, but also look at the EEG signal. So I will first look at the behavior. So what we do here with the noise is this, might be a snippet of what the participant does, and the participant will make some responses. And then I will go in and cut out the noise here in the yellow square leading up to that response. And I will do that for each response, average this, and voila, what I get out is this integration kernel of the average coherence from these snippets. And if we do this for each condition, rare versus frequent, short versus long, we indeed see that the integration kernels vary between conditions. And indeed, if you are in a rare trial condition, you consider evidence further back in time than what you do for frequent trial conditions. So what about the EEG? Sarah already mentioned this. We also obviously know about the centro-parietal positivity signal from O'Connell, and that's based also the reason why we looked at certain electrodes in this case. And again, the noise plays an important role, because if you think about your typical EEG experiment, you might have your 30 to 50 events that you can time log to. But in my event, I basically have thousands of events, because I can look what the brain does at each time point the noise changes. And yeah, I don't use a typical time log analysis. It's more regression-based analysis described in the Unfold toolbox by Inge and Dijmingen. They will give a talk tomorrow at Neuromatch, so check that out. It's really interesting. And what I did is I first wanted to know what happens in the brain every time there is a change in the noise. What's the main effect? And this is shown here on the left. So from the topography, you can see this is sort of the centro-parietal partitive signal that we know from O'Connell and his studies on evidence integration. And you can also see it has a peak at 300 milliseconds after a change in the noise. And this is consistent across all conditions. And I think this is really cool, because you really see, okay, the brain updates or does something every time there is a change in the stimulus. But even more interesting is that if we now look put on top of that requester looking at how much does the noise change, meaning how big is the difference of the previous coherence of the previous jump and the current coherence of the current jump, you can see that we find the difference slightly only for rare versus frequent trials, but this is significant and indicates that if participants are in rare trial conditions, the brain updates the information differently than if you are in a frequent trial condition. We also have some additional results that I think make the previous results even more interesting. So what I also did is I did a control experiment. In this experiment, participants not only see this horizontal motion, but they will also see vertical motion going up and down. This vertical motion follows the same statistics as the horizontal motion, but will give you no indication whether you're in a trial period or not, because the participants should still focus on the horizontal motion. And that's really cool, because now we can see that the brain, actually, we're looking at the same regressors, really only cares about the motion relevant, here the dark gray line, information in the horizontal motion, but it doesn't really care about the vertical motion. So the signal we are measuring is really directly related to decision-making and doesn't have to do with any other processes, such as a visual update or something like that. And another really interesting result, I think, is this one here. So now we are back with behavioral integration kernels, and this is my final result. What we actually found is that the cross-subject variance is a lot higher than the within-subject variance. So what this means is, so these are integration kernels for 3 subjects in the row. And the conditions are here in the columns. So if you look at subject 1, the integration kernel is actually quite consistent across all conditions, but it varies quite a bit across subjects. What I mean by that is, here the integration kernel decays very fast back to the baseline, which means the subject actually only considers the very recent motion directions the subject has seen. And this is true for all conditions. In contrast, if you look at subject 3, here the decay is a lot slower, meaning this subject actually considers evidence that goes way further back in time. And then I guess subject 2 is somewhere in the middle. This means that actually we can get here a robust signal of the integration kernel for each subject, and we are obviously continuing analyzing this now, because we think that we might also find such a signal in the brain that might be consistent within a subject but vary across subjects. So in conclusion, I talked about a new continuous evidence integration task that allows us to measure evidence integration robustly. Furthermore, by manipulating this task, we can influence the behavioral integration kernels participants use, and also the EEG signal. The measured EEG signal is purely related to this decision-making process, but not to another process, as I've shown with the control study. And these behavioral integration kernels can be robustly measured and are consistent within the subject and are further investigated now. Thank you for listening.
That's great. Thank you very much, Maria. Right, and actually quite on time, quite early, in fact. So we've got plenty of time for questions, and they're beginning to stream in now. So I'll just leap in here with the first one. So we have a quick question from Benedict. The effective jump is a constant, and prediction error is the slope. Is that right?
Yes. So the effect of the jump is a constant, so it's a binary regressor for a 1 every time there's a jump. So it's really the main effect size. And the prediction error is really actually the difference. So let's say if the jump at time point t is at 30% coherence and the next one is at 20% coherence, and the difference is 10%, and that would be the regressor. And I've plotted the betas for each of the regressors. Does this answer this question?
Okay, yeah. And the other part of the question was the effect sizes in microvolts.
Well, I plotted the betas, and I think for the jumps, that's obviously true because it's just the main effect. But for the prediction error, I wouldn't be quite sure now. That's true.
All right, so maybe Benedict can follow up by email on that one. Okay, so we have one from Tim Hanks. Very interesting experiment and results. If I understood correctly, your hypothesis was that kernels would be longer for long duration than short duration trials, and bound would change based on the rarity of trials.
Yes.
But it
so
seemed that, yeah, sorry, go ahead.
Sorry to interrupt you. Yeah, no, yeah. So like just nothing. Yeah. So yeah, that's true. You've seen that correctly. I didn't really go into that because so definitely. So the first thing I can say, we so far haven't found any changes in the evidence threshold for which we had hypotheses for rare versus frequent trials, but indeed we found that we have a very strong effect of the trial frequency with rare trials having longer integration kernels. It seems like the participants just need more evidence in these paradigms to make a decision. Sorry, I need to read that. And that they just need, you know, in total higher evidence to be sure that they are in a trial. I should also mention that this also holds for false alarms. So if participants press during the baseline period because they thought they are in a trial, we also find a result that still shows that the integration kernels are still slightly increased for the rare trial conditions.
Okay, yeah. So I think that the next part of the question was, do you have ideas for why that occurs?
So yeah, so I think, like I said, so for the rare versus frequent is really just a matter of how much evidence do I get, and that I really believe that participants need more evidence, because I guess if you're in a rare trial, you need to be convinced a lot more, because trials occur. You're a lot more cautious to press the button, right? So you think that you need more evidence, you need to be more convinced to press the button, versus in frequent trials, they happen so often, like every 2 seconds, that you basically press constantly because you're a lot less cautious about it. And then you obviously would consider probably less evidence back in time. I hope that explains this a bit.
Yep, I think so. Cool. So we have a question from G1. Interesting talk. What is the reason that you looked only into certain time before the button press, not the entire period of a trial?
Okay, yeah. So the thing is that, you know, we have these 5 minutes. That's one block of one condition. That's condition in that block. And we always have trials dispersed in there every so many seconds, depending on the frequency. And these trials are either 5 seconds long or 3 seconds long. So that's why we look 5 seconds back in time.
Okay, great. All right, from Maxine, who says, a lovely talk, thank you. And in a recent paper from the Shadlin lab by Gabriel Stein, The authors pointed out that evidence integration over time is an assumption that we need to test against alternatives such as extremer detection. Have you tested whether people are indeed integrating continuous evidence?
So I would argue, given that well, yeah. So the thing is that we only work with an on-site Boolean stack process so far to model this task. So it sort of to some degree overlaps with the results I found. But I think we are now also looking into more of like a Bayesian model, which will Lillian Weber, a postdoc with Lawrence, do now, where I guess we will also look further on how each jump directly influences the evidence integration signal and how much the variance of the noise influences how you integrate that evidence.
Okay, great. All right, so there was some remaining questions, but I think we're going to have to move on to the next talk now. So if any of the remaining questions, you could maybe have a look in the chat and answer them, and also maybe people can follow up with you by email as well.
Sure, and my email address is down there. Thank you.
Right, and now the final talk of the session is.